What do you think is the absolute simplest task in a laboratory? I'd bet it's something like making a 1% sucrose solution, right? It's just sugar and water. But what if I told you that the incredibly rigorous process behind this one simple mixture contains the entire blueprint for every single trustworthy scientific discovery ever made?
Okay, stick with me here, because we're going to dive into this. Understanding how to do this one thing perfectly, and, more importantly, prove that you did it perfectly, is the secret key to separating real, actual science from just random noise. This little flask of sugar water, for us, it's going to be our model for understanding the very foundation of scientific trust itself.
You see, to get results that anyone can trust, whether it's the person at the next bench or a lab halfway across the world, science can't just be a bunch of creative, random steps. Nope, it needs a strict, universal rulebook. And in the world of non-clinical research, that rulebook has a name: Good Laboratory Practice, or GLP for short.
And what's so wild is that this whole complex system really boils down to one incredibly powerful idea. This quote, this isn't just some catchy phrase they hang on a lab wall. It is the absolute beating heart of GLP. It means every single action, every measurement, every tiny observation must be documented. You have to create a permanent, traceable record. Because without that written proof, your work, scientifically speaking, just didn't happen. It's like it never existed.
So here's the formal textbook definition. But don't let the words trip you up. Just think of GLP as the quality control system for a laboratory. Its entire purpose, its whole reason for being, is to guarantee that the data being produced is reliable and, crucially, that it can be reproduced by other people. It's the framework that makes sure the final results are built on a foundation of solid rock, not sand.
So how does this framework actually work? Well, it's held up by 5 key pillars. First, you've got standard operating procedures. We call them SOPs, which are basically the written instructions for how to do literally everything. Second, documentation. That's the proof that you actually followed the SOPs. Third, you need to calibrate your equipment to make sure your tools are telling you the truth. Fourth is traceability, so you can follow any result all the way back to its source. And finally, proper training, to make sure everyone on the team knows the rules of the game. Put them all together, and you have a real system of discipline.
All right, so we've got the rules of the game. Now let's talk about the tools. Because following the rules is one thing, but you also need the right concepts and the right equipment to play properly. This is where we get into the real nitty-gritty of generating data that you can actually trust.
First up is a word you've heard a million times: accuracy. And this one's pretty intuitive, right? It just means being correct. If the true value is exactly 10 grams, an accurate measurement is one that gets super close to 10 grams. If you're playing darts, accuracy is hitting the bullseye. Simple as that.
But now we have precision. And this is different. This distinction is so critical. Precision is all about repeatability. If you take 3 measurements and they are all incredibly close to each other, that's high precision. Back to our dart game, this is like throwing 3 darts that all land in a tiny little cluster over in the corner. You're super consistent, but you're not necessarily correct. And of course, what we're always aiming for in good science is to be both accurate and precise.
And this difference perfectly highlights the 2 kinds of enemies a scientist is always fighting. Accuracy's enemy is systematic error. Think of it as a consistent flaw, like a scale that's miscalibrated and always adds an extra 0.1 grams. Every single measurement is going to be off in the exact same way. Precision, on the other hand, its enemy is random error. All those unpredictable little things, like a tiny draft of air hitting the scale, or you slightly misreading a line on a beaker. These errors make your results scatter all over the place.
And believe it or not, controlling these errors comes down to choices as basic as the containers you use. When you need the absolute highest accuracy for making a solution of a precise concentration, you have to use what's called Class A volumetric glassware. It's non-negotiable. But for other jobs, maybe in biology where you need to throw things away to avoid contamination, plasticware is the smarter choice. It's all about strategically choosing the right tool to minimize error for the specific job you're doing.
So let's bring all this back to our humble sugar water. The entire detailed procedure can really be boiled down to 4 critical phases. 1. You weigh the sugar precisely on a calibrated balance. 2. You dissolve and transfer it, making absolutely sure every last grain gets into the final flask by rinsing everything. 3. You dilute it perfectly to the calibration mark, reading that little curve of the water, the meniscus, at eye level to avoid any visual errors. And finally, 4. You homogenize it, turning the flask upside down again and again so the concentration is perfectly uniform. Every single step is a deliberate defense against error.
Okay, you've done it. You followed the rules, you used the right tools, you were meticulous. You have your data. But guess what?
the job isn't over. Not even close. Now comes the final and maybe most crucial stage, proving that your results actually mean something. Welcome to the judgment of statistics.
This, right here, is the fundamental question that statistics was invented to answer. Was that change you measured a real effect caused by your experiment? Or could it just be a fluke, a result of all those tiny random errors we were just talking about? Honestly, without statistics, you're just making a guess.
So the first thing you have to do is just look at your data. What's its shape? Does it all cluster nicely in the middle and tail off at the ends like that classic bell curve you've probably seen? If yes, great. You get to use a set of really powerful statistical tools. But what if your data is all messy or skewed over to one side? Well, then you need a different, more flexible set of tools. It's like choosing your wrench. You have to use the right one for the job or you're not going to get a valid result.
Now, to test their data, scientists do something that sounds a little backward. They start by playing devil's advocate against their own ideas. They create something called the null hypothesis. This is basically the most boring default assumption you can make, that nothing interesting happened, that there's no real difference between your groups, and any little changes you saw were just due to random chance. Your entire goal as a scientist is to gather enough evidence to be able to reject that boring explanation.
And the single most important piece of evidence they use to make that decision is a little number called the p-value.
The p-value basically tells you the probability that you would see the results you saw if that boring null hypothesis were actually true. And in many, many fields of science, the gold standard, the magic number, is a p-value of less than 0.05. This means there's less than a 5% probability that your findings are just a random accident.
And when that p-value drops below that 0.05 threshold, well, that's the moment of truth. That's what gives a scientist the statistical confidence to stand up and reject the null hypothesis, to say, no, this was not just random noise, and to declare their findings statistically significant. This is the moment a bunch of careful observations officially gets to graduate into being a potential scientific discovery.
So, let's bring this all home. We've gone from simply weighing sugar all the way to some pretty complex statistical ideas. But why? Why does this incredibly meticulous, you might even say obsessive process matter so much? Because it builds an unbreakable chain of trust. Look at how it all connects. It starts with a meticulous procedure. That procedure is locked in place by the GLP framework of documentation. It's carried out using accurate and precise tools. The results from those tools are then put on trial and validated with statistics. And all of that together leads to the final prize, a reproducible discovery that another scientist can trust and build their own work on top of. If any single link in that chain breaks, the whole thing falls apart. And that really is the ultimate point. This whole structure of rules and tools and validation isn't just about being a perfectionist. It's what allows science to be a global collaborative effort. It's what allows a researcher in Tokyo to completely trust and then built upon the work of a researcher in Buenos Aires whom they've never even met. This is the very foundation that all of our scientific knowledge is built on. Which kind of leaves us with one last question to think about. We always celebrate those big, flashy, eureka moments of discovery, right? But maybe the real genius of science isn't found in that final flash of insight at all. Maybe it's found in the incredible, unseen discipline of the thousands of tiny, perfect steps, like just making a simple solution of sugar water that made that insight trustworthy and ultimately true.